this afternoon's topic is epigenomics. So I actually like that term, epigenomics, because it reminds us that it's not really epigenetics. So epigenetics classically is non-Mendelian inheritance. So inheritance due not to genotypic variation, but to modulation of the genome. Then over time, the term epigenetics came to include somatic alterations. So changes in cell states that altered the activity of chromatin. Such that the dynamics of DNA function were altered in a manner that was transmitted through cell division somatically. And so epigenomics, I think, captures that notion much better because it's really not talking about the genetic consequences so much as about really profiling what's going on in the genome to modify the DNA and to modify the factors that interact with the DNA and how they then impact gene expression and how they help us to interpret what regulatory DNA variants are doing. So I hope most of you have heard of this thing called the ENCODE project, which stands for Encyclopedia of DNA Elements. This was the first main paper that came out in 2016. In fact, it was one of these papers that came out across many, many journals, and you can look up a compendium of results. They've done the same thing a couple of years ago with phase 2. And then there's also something called the Roadmap Epigenome Project from John Stam at University at UW in Seattle, which is doing parallel sort of work. And what the ENCODE project is doing is trying to annotate biochemically what's going on in chromatin throughout the genome, and then from that to try to infer function. Now, those of you who were in the previous module would have heard Joe Lachance mention that Ewan Burney, one of the leaders of this project in Europe, estimates that 80% of the genome is functional because something like 80% of the genome has differences in the chromatin across cell states. We both think that there's a little bit of a stretch to actually infer from that 80% of the genome is functional, but nevertheless, that's what you observe. So this top figure here is really sort of showing many of the different things that can happen. Okay, so high C, or CHIA-PET, but mostly these days high C, are assays that allow you to look at long-range interactions between elements in the DNA. So typically long-range means within a megabase, but you even do also get long-range interactions across chromosomes in some cases. So what Hi-C is doing is actually taking advantage that pieces of DNA that make contact with each other. So there's a looping out of the intervening DNA. Excuse me, those bits that make contact, you cleave the DNA around them and then religate it together. So the bits that were brought together actually now covalently are linked to each other in a manner that allows you to be captured and then sequenced. So Hi-C is very, very useful for actually determining which polymorphisms and which open chromatin segments make contact with the promoters of genes. And if they do that, then actually it's a pretty good sign that they're probably an enhancer or a silencer for that particular gene. I just used the word open chromatin. So this next set here, DNA-seq, FAIR-seq, and ATTAC-seq, are all methods for actually annotating pieces of chromatin that are not as tightly wind up in histones and coils and topologically active domains as others. And so they're open to digestion with DNAs or to attack by transposons, as we'll see in a minute. And you can use that to very accurately estimate the tens or hundreds of thousands of places in the genome that are open in any given cell type. ChIP-seq refers to chromatin immunoprecipitation sequencing. So if you want to find out where any particular transcription factor is binding to the genome, what you do is you mildly cross-link that protein transcription factor to the chromatin DNA, you precipitate that, and then you sequence the DNA that was bound to those. So that's a very good way of actually finding out where in the genome there is DNA bound. And it doesn't just work for transcription factors, you can also do it for histones or particularly modified histones. And you can use that then to identify where there are particular classes of active histone. So histones are these sort of octuplets of proteins around which DNA binds tightly under normal circumstances, separated by short stretches. So almost all DNA is tightly wound up, so that it fits inside the nucleus in a tight manner. And then when it's the DNA is being transcribed, it's open for business. Usually there's an unwinding of those histones. Part of the thing that modifies histone and transcription factor binding is the methylation state of the genome, as we'll see. So whole genome bisulfite sequencing, reduced representation bisulfite sequencing, and methylation arrays are all different ways that we'll briefly talk about of measuring where in the genome there's methylation happening. So that's a very good mechanism for imprinting genes or for opening and shutting genes. As we age, we get more and more methylation, and actually you can use total methylation levels as a measure of somatic aging. And then we've talked about RNA-seq. There's a whole area of bioinformatics that's just based on computational analysis of DNA sequences. And then there are things like ClIP-seq and RIP-seq that actually measure ribosome-bound transcripts, for example. So you know which ones are not just which transcripts are there, but which ones are actually being translated. Okay, so these and other methods constitute the cornucopia of ENCODE assays, and now initially they were assayed in 4 major human cell lines, but now you have model organism ENCODE, you have about 100, 150, I think it is, less well characterized cell lines, you have tissue models, so it's a real growth industry. And the importance of it is just, you can look at this in more detail yourselves later, but basically it turns out that regulatory variants that come up in GWAS, so the sites in the genome that regulate complex traits, are all targets of these assays. They're very heavily enriched for these assays. And we'll see a little bit of that in my last lecture tomorrow. Okay, so let's start with DNA's hypersensitivity analysis, which these days has actually pretty much been replaced by ATAC sequencing. But the way DNA's hypersensitivity works is that DNA is not protected by binding to a protein, then it's accessible to this enzyme DNase. And DNase will come in, it makes a double-stranded break. So then you just ligate adapters to that. You can pull down those adapters through something called streptavidin, which binds to biotin. You just use a magnetic bead to pull those down. You ligate sequencing primers on them, amplify them, and then you just massively parallel sequence. And so wherever you get a sequence read adjacent to one of these cut sites where DNA's hypersensitive is, then you can infer that there's open chromatin there. The only difference, well, the major difference with ATAC-seq is that instead of using the enzyme DNase, what happens is that this transposon, TM5, is used to jump into the DNA. So where it jumps in, it basically tags that, and then you sequence adjacent to it. The ATAC-seq is just a lot easier to do, and it can be even done in a single cell assays these days. So that's what I'll talk about in more detail. But either way, if you actually sequence to a large enough depth, you get nice peaks. So you get enrichment of DNA in a stretch that might be 40 or 50 bases long. So here's or even longer, up to 200 base pairs. So here's a fairly typical peak from one of these cell lines, K562 cells. The core of that is about 200 base pairs. That's from ChIP-seq. It tends to overlap with the DNase-seq. So this is that transcription factor from the ChIP-seq assay. This is the open chromatin region from DNase-seq. And if you have high enough depth, you actually can find out that right in the middle of that peak, there's a protected region. So that's where actually the transcription factor itself binds, and it protects that from the transposon or from the DNase because it's bound to the DNA there. So what happens is that the protein binds, it protects it, but for 100 or so bases on either side, it becomes open and accessible. And then it gradually or quickly decays away to sort of background levels of protection by histones and things, okay? And then the other thing that this slide is showing is that those sequences which are bound by transcription factors are highly conserved because that's, so natural selection ensures that transcription factors that bind to these sites, that the sites themselves are conserved so they can bind there. So you can actually measure vertebrate conservation if they're important sites. And what we're seeing over here is just a great example of this, that this is the actual profile of DNA is hypersensitive. So there's a protected block. And then right next to that is there's an intense peak, and then it decays away over about 80 base to 100 base pairs on either side. And then if you look at the levels of vertebrate conservation, it's highest in the middle of that binding site. So in this case, it's CGC and TGCGC. And it turns out that those sites, if you look in the same places in the genome, the intensity of both of these signals are proportional to the intensity of the ChIP-seq binding. So it doesn't always work that nicely, but that's the sort of ideal picture that you want to see. And so what it's telling you, if you do this, is you've got open chromatin. You can find out the motif that's there, so you can infer what transcription factors available. If there are databases you can look up, you can actually confirm that that's the binding site. And then you can look up polymorphisms in those binding sites and make inferences about those variants being functional in affecting the target gene expression. And all of this gets annotated in things like UCSC browser. So there are tracks in that browser that have ENCODE or roadmap epigenome data where you can look up all of those DHSs and ATAC profiles in the cell types of interest. And that's really useful, very, very cool bioinformatics. So we've got an assay in our lab right now using CRISPR to try to identify variants that influence gene expression in single cells. And generally what we find is that the sites that give positive results in our assay invariably overlap with one of these ATAC peaks that we can look up in the database. It's very, very useful. Okay, so the basic tools for doing ATAC-seq or statistical tools are very similar to what we have for gene expression profiling. In fact, EDGE-R and DE-seq2 are the most commonly used software, or until recently have been the most commonly used software for doing differential accessible region analysis. So instead of differential expression or DE, we refer to differential accessible region. And ACR is another acronym, stands for accessible chromatin region. Some papers you'll see them called open chromatin regions. The basic idea is that accessible chromatin is identifying enhancers or promoter sequences that are active. So what peak calling code is doing is looking for an excess or an enrichment of reads against the background of the DNA, indicating that there's a peak. And the most commonly used codes for doing that is something called MACS2, MACS2 there. But there are a couple of major differences actually to RNA-seq. So one is that if you think about what's going on in a single cell, that accessible chromatin actually at any given nucleotide or any given binding site basically needs to be coded 0, 1, or 2. So either it's not accessible at all, or one of the two chromosomes, the pair of the diploid pair is accessible, or both are accessible. So at the single cell level, even though you may have PCR amplification, ultimately what you're getting per chromosome is a 0, 1, or 2. So single cell, that's what you're measuring. And in bulk data, of course, you've got a distribution of cells in either state. So it's more of a quantitative measure, but it's very, very clearly negatively binomially distributed data. So whereas a small fraction of transcripts are negatively binomially distributed, a large fraction of ATAC peaks are that way. And so this is a nice review that came out a couple of years ago in Scientific Reports, Gontaz et al. And what it's showing is that if in blue, this is now on the log scale, so log base 10 scale. So counts per million above 100 is responsible for maybe, well, so a quarter of the genes are in that range. Then a huge number are in this range of 10 to 100, and then you've got a tail of low abundance transcripts. In ATAC, if you do it on the same scale, you actually find that 50% of the reads have 0, 1, 2, 3, or 4 counts per million, so very, very low, and about 25% are in this range, which would be regarded as low in RNA-seq. So distributional properties are really quite different, and then that's going to affect the test statistics and the properties of those. And then this plot beside it is sort of showing typical variance around those intensities, so they're fairly tight. So if you're looking for a 10% mean difference in, between two treatments, then you need to see that distribution being quite small, 20%, 50%, and so forth. So actually you do have quite a lot of power for seeing this, because actually the variance within single cells is, within a tissue is quite low. All right, so the workflow, there's another really good review that came out around about the same time. Part of the title is A Hitchhiker's Guide to ATAC-seq Data Analysis. So if you really want to learn how to do this, then that's a fabulous starting point. Is Yan et al. 2020 in Genome Biology. And they go through the steps of ATAC analysis. So it starts with quality control, which is divided into two phases. So there's pre-alignment quality control. So the first thing is to make sure your reads in your library are correct length. You want to make sure that they're not biased with respect to GC content. And so that's actually fairly standard in sequencing basic sequencing software. Like Cell Ranger, for example, you can get that. And then you also want to check there's not a large number of duplicates. And then once you've got that data, you then align it to the genome, just like you would for RNA sequences. And then one of the unique assays you do is look for enrichment near transcription start sites, okay? So ATAC peaks can either be right adjacent to the transcription start site, so in the promoter of a gene, where the histones have been lost, and they tend to be large nucleosome-free regions, or they can be an enhancer, so they could be anywhere up to a megabase away from the transcription start site. And they're maybe about 50-50 distributed, so what you're looking for is enrichment in the TSS. So if you don't see that, it was probably a bad quality library. So the images over here are just telling you that ATAC-seq started to displace, well, this whole field started about 2016. You got a lot of papers doing DNA-seq, and then ATAC-seq came on pretty quickly. And so by 2018, 2019, it was the vast majority of these sort of profiling. Panel B is just showing you the fragment length distribution that you get, and it's bimodal. So you get fragments in the range of about 80, and then fragments around about 200. So these ones are bound to nucleosomes, because there'll be a nucleosome in between two regions. And so that's typically enhancer-like, and then this would be what you'd see in promoter regions, shorter fragments. So you see those, and then you can map where those are relative to the actual inferred start site of transcription. So you get sites both upstream and downstream of the promoter. So that's how they map. And then if you ask where they are, about a quarter of the reads for this particular cell type are in the promoter, about a third are distal or intergenic. You get about 25% are intronic, or a third, well, 10% in the first intron, and others in other introns, and then you get other regions. Okay, so most of the reads are in the vicinity of the gene, about half, and then about a third to a half are intergenic in regulatory regions. Okay, so once you've done that, the analytical steps are to call the peaks, okay, using MAX2, evaluate differential accessible regions using similar statistical tools as the ones we've talked about, then try to annotate the genes that are in the vicinity of the peaks. Some software lets you try to predict the expression of those genes based on the open chromatin. Very often you want to find out what motifs are enriched underneath those peaks. More and more, the field is moving towards multi-omic integration. Okay, so integrating ATAC data with transcriptome data, with high-C data, with chip data, so all putting that together. I'm not going to talk about that. It's still a maturing field. And then ultimately what you want to get to is gene regulatory network inference. So that you can sort of say, well, this chromatin's open. It's typically bound by this transcription factor. When this transcription factor gets turned on, we see those peaks closing. And so you start generating models as to how it is that these changes in gene expression of regulatory molecules are causing downstream consequences in the cells. So it's becoming more and more reductionist in that sense. Okay, so the next slide here is talking about hypothesis testing for ATAC-seq data. This is from that same review I showed earlier, Gontaz et al. And so the first point they make is that the very high percentage of zero counts, so that negative binomial skew makes t-test inappropriate. And in fact, it turns out, at least for small samples of just half a dozen samples of each type, that Wilcoxon rank sum tests, which we use commonly for transcriptome analysis in single cells, are actually very inappropriate for ATAC data. So that's shown down here. So what they've done is they've compared rank sum to t-test, to EDGER's basic workflow, to Limmer's basic workflow, DE6, so two versions of DE6 workflow. And this is just the recall. So the true positive rate. So how good they are at identifying ATAC-seq peaks defined by a gold standard, really deep analysis of bulk tissue. And so what you see is that t-tests don't do particularly well. DE-seq2 is better. EDGER and DE-seq are performing somewhat better than that. And then Limmer at 1 count per million, so very, very low abundance peaks. So that is a small fraction of the cells open. Limmer's best, but as you get up to 5 or 10, so counts per million for those sites, all of those tools, with the exception of t-test and Wilcoxon rank sum, work pretty well. But then down here, what you're seeing is that if you also ask it to have a low false positive rate, well, actually the false positive rate is reasonably controlled, so it's less than 5% for all of these tools. But what you see is that it's a little bit higher for Limmer and the t-test. And then it goes down for DE-seq. In fact, DE-seq2, they argue, is the best. So if you want to control both the false positive rate and the sensitivity, or retaining sensitivity, Limmer's probably the best, or DE-seq2. Actual power, of course, is a function of the sample size or replication rate. So here, what they just modeled was n equals 3 per group. But they go on later in the paper and say that once you get past n equals 15 samples per group, that actually Wilcoxon rank sum and t-test do much, much better. And in fact, they're recommended, sequencing depth is the other major factor, and they recommend having at least 20 million ATAC reads per sample. So actually less than transcriptome, in part because it's not dominated by this big tail of a few genes contributing most of the reads. So you can get away with about 20 million, not just get away with, that's what's recommended, is about 20 million per sample. And then they had a few more analyses in this. So that was all based on simulation studies. And they went and actually confirmed this with some real data of a comparison of liver and kidney. And so the principal components we talked about before the break, in this case, 98% of the variance in the ATAC peaks is between liver and kidney. So in other words, there's a big difference, many, many thousands of sites differentiating those almost perfectly. In fact, there are about 30,000 different ATAC peaks that separate those two tissues, and the vast majority of them are in common between the three methods. So that's good news. The three methods are preferable in this real data at the stringent cutoff of p less than 0.1 with a fold change greater than 2. The second plot over here is actually showing that sensitivity remains high. Also in this data set for Limmer and EDGER, it starts to drop off for DE-seq2. So what they're doing is downsampling. So they started out with 6 replicates of each of the 2 tissues, and then they just removed one, then removed another one of each, and 2 of each, 3 of each, 4 of each. And what you end up with is pretty robust statistics, down to even as few as 3 replicates for recalling DARs under this threshold. Now, of course, you at this point should be saying there's nothing magical about a 2-fold change, and that's true of ATAC-seq data as well. There's also nothing magical about a p-value of 0.01. In fact, we recommend a false discovery rate criterion. So this particular volcano plot, instead of plotting the p-value, is plotting, I think, the Benjamini-Hochberg FDR rate. So all the sites above this, 99% of them are true DAR by significance levels. And then this is like a 50% fold change, so 1.5 fold change. This is a 2-fold change out here and reciprocally. And what they're doing and showing in this particular plot here is to ask the question, which one is a good cutoff? They actually compared the ATAC signal at those 30,000 sites with the differential expression between liver and kidney at the genes linked to those sites. And what you're seeing in the green and the red or orange is a very good correlation, but that's much lower if you go to the blue. And so the blue is if you include all the significant sites, and the red and the green, what happened if you get the 1.5-fold or the 2-fold. So their argument in this paper is that actually 1.5, some combination of 1.5-fold difference and FDR less than 1% is a very nice way to identify ATAC peaks. And I really wouldn't quibble with that. That's sort of fits our intuition and our knowledge. Now let me go and see what's in the chat room here. So the term, thank you, Justice. So the term hypothesis testing just refers generically to the question you're asking with the data. So in this case, the hypothesis, the null hypothesis, is that there's no difference in ATAC profiles between liver and kidney. The alternate hypothesis is that they're either up in the kidney or in the liver. So that's a very, very simple one-way hypothesis test. Okay. So when I use the word hypothesis test, what I'm referring to is a statistical evaluation of a contrast between two or more conditions. And when I go ahead and forget to turn on my pointer, remind me, please. It's too late now. Let me get my pointer on. Okay. All right. So there's a bunch of software for doing ATAC analysis, and there is a large number of papers that evaluate that software. So one of the first ones to come out was Chen et al. In genome biology, and that one recommended something called SnapAttack, single cell analysis. Those of you familiar with Seurat for doing single cell transcriptomics, it has its equivalent is something called Signac. So Paul Signac and Paul Seurat were the two pointless painters, where the name comes from. There's another version, which is one that we use in my lab called Archer. That's from this group at Stanford, Howard Chang's group, Bill Greenleaf's group at Stanford. In fact, the first author, Runciman Satpathy, is now also a faculty member at Stanford, junior faculty member. And this is self-serving because it's a table from their own paper, this Nature Biotechnology paper. But it's sort of comparing the features that they think are important that you want in analysis software. So is it easy to import the data? And actually for Maggie Brown in my group, who's the main one doing ATAC analysis, for her, that was a really big step. So actually importing data into SnapAttack was not easy, and Signac, not easy at all. They've all got similar QC metrics. I think they even use the same code. They've got, Archer has a doublet removal. So doublet refers to cells which appear to be made up of two cells. That's certainly going to be in new versions of those softwares. They have a tool called Magic, which imputes gene expression. I'm not so keen on it, to be honest. They have dimensional reduction that Blake Pong just spoke about. Usually it's done with UMAP visualization. So actually the clustering is a separate step. So UMAP and t-SNE are methods for visualizing data. Peak calling, motif enrichment analysis, estimation of which genes are nearby, footprint analysis, data visualization tools, trajectory analysis, all of these things are implemented in most of this software. But as I said, this is trying to argue that Archer is better, and we've actually found it's very useful in our analysis that we're doing of single cell ATAC data. So here's the workflow. So you start out with peak calling, you search for motifs and footprints, maybe overlap with variation in the chromatin. You project bulk ATAC data onto your single cell just as a reality check. Sometimes you visualize the tracks, create a genome browser for your own group to do that. You try to predict gene activity. Peak co-accessibility analysis is shown over here. So I don't think I can blow this up. Let me get out of me and show. Let me see if I can blow this up a little bit. So what you see is you visualize your different cell types. So this might be a single cell analysis, and you've got 20 different immune cell types, for example, in the analysis. And each of these sort of blue peaks here is literally an open chromatin peak. So you can see that these ones here are in common. They're in the promoter region in all of the different cell types for this particular gene. And then this is a less strong peak, but it's also in all of the cell types, maybe the exception of one. But these lymphoid cells all carry that peak, whereas this other type of cells down here don't. The same here. So then you can ask, well, across all of the cell types, which ones are co-regulated? So they draw these plots here. And on the basis of that, you can go ahead and try to create gene models. So you weight those and you try to get a gene activity score, which is basically predicting expression from the whole profile of ATAC. I think there are more sophisticated ways of doing that. We and other groups are working on those, but ultimately what you're doing is moving from ATAC to predicted gene expression. However, that's really not the main utility of this. I mean, if you're going to try to predict gene expression, just measure gene expression. What it really is for correlating which peaks, which open accessible regions are the ones responsible for the gene expression. And there are plenty of examples where actually the two measures are anti-correlated. CD19, for example, is one that I'm particularly interested in, where the loss of important binding sites correlates with activation of gene expression because the regulation is negative. And so you have to be aware of that an open peak does not necessarily mean an active gene. So it's very, very important to model that thoughtfully to sort of move from open peak to predicted gene expression. Linking peaks to genes can either be done by correlating expression and ATAC, or better is this integrative genomic process of actually looking up high C profiles to find out which gene that peak links to. Is there a way to perform peak identification on ATAC scores accessible in a browser? Let me get back to Maggie and let her give a good answer to that question. She's more, that's a question for somebody doing it practically on her own. Let me get back to you on that, okay? All right. Okay. Yeah, and then there are all sorts of other things. So there's trajectory analysis that Tom will talk about with RNA. There's trajectory analysis you can infer from ATAC, and all sorts of other things. There's the marker identification can be useful. And then the state of the art right now is what's called multi-omic analysis. So that is where you measure ATAC and RNA on exactly the same cells. So up until now, you've basically taken your cell population, divided into two, and done the assays on them separately. And when you do clustering on those, you get some correspondence, but it's never perfect. If you actually do it on the same cells, then you know that the cluster, you can use the cluster, you do joint clustering, and you can infer so much more. So multi-omic analysis is literally the same cells having both measures on them. So very, very powerful. So that's where the state of the art is. All right, so that's ATAC-seq. That's all I wanted to say about that. The other major type of epigenetic regulation that people care a lot about is methylation, okay? And really there are two types of methylation. So one is methylation directly of the DNA, and the other one is methylation of the histones to which the DNA binds, okay? So it turns out that there are different types of histones, and there's a code for this. So histone 3, if it's methylated 3 times at the 4th lysine, then that's a particular chromatin mark, and that tends to mark active genes. I say tends to because there are always exceptions to that, but it's generally a marker of active genes. Whereas if lysine 27 is trimethylated, then that's associated with repression. Okay, so people are very busily trying to work out this histone methylation code. But the way you analyze this is through that ChIP-seq assay. So you actually have antibodies that recognize the methylated forms here. You use a chromatin image precipitation to pull down histones that bind to that antibody, and then you sequence the DNA that they will cross-link to. That's different from methylation analysis, which is identifying sites in the genome that are methylated. I'll show you how in a second. But an important point to note here is that there are opposite results between the open upstream regulatory regions and the gene body, the coding regions. So typically methylation in the gene body is associated with repression of transcription, whereas methylation in the upstream regulatory region tends to be associated with activation of transcription. So you have to know where in the gene model you're looking at. There's a terminology that is CPG islands. So it turns out that methylation happens at CPG or CG dinucleotides. So the P stands for the phosphate linkage between them. And that it tends to be groups, clusters of CPGs in a row all get co-methylated. So we call them CPG islands, and they're often next to promoters. And then as you move away from those islands, you get what are called shores, just like the beach that I've just been walking on. And then you get into island regions. They're not called anything. And so you can look up in databases what the standard methylation levels are in each of those and what it means for transcription. There are genes that buck these trends, but for the most part, that's the general rule. High methylation in CPG islands upstream of TSS is associated with open active with active transcription, whereas high methylation in gene bodies is the opposite. The other type of assay that I've mentioned is high C and chia-PET. So the idea is that a red upstream region is brought proximally physically inside the nucleus close to the promoter, represented by a green region. You cleave those with a 4-cutter enzyme and you religate so that with some probability they ligate back together. Here's a different site doing the same thing. And then there's ways to pull down those ligated products, and then you sequence them. And so if you see that the same read, same paired end read has sequence from a promoter and somewhere else in the genome, that somewhere else must make proximal contact with the promoter. So the most famous example of this, practical example of this, is dissecting the function of the FTO gene. So FTO is one of the largest effect polymorphisms in the human genome. Individuals homozygous for 2 different alleles at FTO differ by weight by as much as 2 kilograms. Actually, I think it's 2 kilograms per allele, so 4 kilograms on average. So it's a really large effect allele. And it's located just upstream of the FTO gene. But it actually turns out that even though there are promoter contacts between the enhancer and the promoter of FTO, there are just as many, if not more, promoter contacts to this gene IRX3. And then there's one called IRX5. And both of these are expressed in neurons, they're neuronal markers. And so people now think that the activity of FTO is not so much mediated by the closest gene, is actually mediated through this more distal gene, which is active in affecting neuronal function, possibly affecting satiety and how much we eat. At least that's the current model. In fact, it's clearly more complicated than that. There's subsequent papers that shown that you get slightly different peaks in different tissue types, so it's probably a complex pleiotropic effect where this enhancer is actually operating on FTO, IRX3, IRX5, and maybe some other genes. Okay, but nevertheless, this assay is a very, very powerful one for helping us see which gene or genes are acted upon by an enhancer. And I might as well say at this point as well, it makes an important point that you cannot infer that the closest gene is the target. In fact, by now we sort of realize that at least half the time, it's not the closest gene, it's some other gene elsewhere. It doesn't even have to be in the same LD block that the region is acting through. Okay, so it takes a lot of care to really put SNPs together with targeted genes that they act through. Right, so how is methylation profiling actually done? Well, the assay is something called bisulfite conversion. So if you have chromatin you've isolated from a nucleus, and you have a set of CPGs in the vicinity of each other, if you treat that DNA with something called bisulfite chemical, or a chemical containing bisulfite, then the protected methylated ones are unaffected, whereas non-methylated Cs get converted to uracil, and upon PCR amplification, uracil is then read as a T instead of a C. So actually what you create de facto is a polymorphism, and that can either be read on an array. So there are methylation microarrays. I think the largest one now has about 800 SNPs. Well, effectively SNPs, they're not SNPs, they're CPGs that you can read. Or if you don't want to do that, you can either use sequencing, whole genome sequencing now. After bisulfite conversion, you compare that to without bisulfite conversion, and wherever you see a C substituted with a T, you know that there was no methylation, whereas if the C is preserved, you know that it was a methylated site. And so it's thought that there are about 36 million CPGs in the whole genome. There are now ways of reducing the representation of those, just to pull down the enriched regions to reduce that analysis to about 3.3 million, which makes the costs more like $500 per sample instead of a few $1,000s per sample. Okay, but all of those three methods are used. And there's actually software that's specifically being written that makes adjustments for methylation analysis and correlation structure, which I should have written down here. I forgot to put it on there. But yeah, the basic ideas of it are similar to transcriptome profiling, but there is dedicated CPG analysis software, which I will put in the chat window later. And just to give you one idea of the sort of analysis that can be done with this, this is one of my favorite papers from one of my graduate students, shared with Subrika Gathasan, a guy named Harry Somaneni. So this was the main paper in his PhD thesis. And what he was asking was whether or not methylation of blood could be used as a biomarker for disease progression in Crohn's disease, inflammatory bowel disease. And it turns out, it's not a good biomarker, but it is a great biomarker for inflammation. So here are just 5 examples of CPGs in the vicinity of 5 key genes that are all involved in inflammation. And what you see is just these violin plots here, or with whiskers, but basically the proportion, so beta is the proportion of sites that are methylated, is high in the gene body of these genes. And the first one, the gray one, is control, so unaffected. The red one is at diagnosis, so still clear change in all of these genes. And then the others are showing that at follow-up about a year later, it doesn't matter whether you're on a biologic therapy, so TNF, anti-TNF, or if you're on an immunomodulator, or if you were on both, or you had some other treatment. In all cases, there was reversion to normality. So actually what's happening is when you're first diagnosed, the inception of the disease, you've got a very inflamed state, and you can see that in the methylation profile that came up. And that involved hundreds and hundreds of these sites. Then he went on and said, well, is there any evidence that methylation is causing disease, or is it responsive? So this is somewhat similar to a Mendelian randomization framework, but basically the idea he tested here is the SNP mediating an effect on the disease through CPG methylation, or is it that the CPG methylation is mediated by the disease? Okay. So what he did is he plotted the predicted effect under this model of beta 1 by beta 2 against the observed effect, which is just a direct SNP to Crohn's disease, and saw no correlation whatsoever. Whereas where he did it the other way around, of direct effect on CPG, modeling the effect of being mediated through Crohn's disease, he sees a very, very high correlation at inception. So in other words, consistently illustrating this idea that what the disease status, which in fact in this case is actually inflammation status, is driving the methylation profile. And then he goes on to show that that disappears upon recovery, either naturally, either spontaneously, or because of biological therapy. So lots of really interesting things you can do with methylation analysis. I should tell you that if you look actually in the bowel itself, in gut biopsies, you get the opposite result. So so other groups, several groups now have shown that in the affected bowel, you do get methylation that persists over time. It's not necessarily inflammatory, and it's probably a decent biomarker of disease progression. And there's also a vast literature sort of arguing that in many cases, the methylation is causal in the change in gene expression. But I think there's an open question on that, actually. I think as I look at data, there's actually some studies have got pretty convincing evidence that methylation is also responding to gene expression. And in fact, for some time, there's been a model that what methylation does is entrenches the chromatin state. So a gene gets activated, chromatin opens, makes it accessible to the methylation enzymes, and then that methylation status makes that open chromatin state persist. But I think we'll get to the bottom of it in many, many cell types in the coming years. So there are some definitions there that I think are important. I'll let you go through most of them. So genome-wide association study is relating a SNP to a trait. So that's just basically asking, is there a difference in disease status is there a difference in disease status or a continuous trait like height or BMI between individuals with the different genotypes at a locus? So if you find out that the allele frequency of AA homozygotes is 20% in cases, but 22% in controls, whereas the, say, GG homozygote is, say, 35% versus 30%, then once you crunch the statistics with very large sample size, that would amount to genome-wide association. So a few percent change in allele frequency between cases and controls is a sign that a SNP is associated with the trait. A TWAS is an interesting modification on this. So there what you do is you generate a model that predicts gene expression from the data. So you have what's called an EQTL study. I'll discuss that more tomorrow. But you can, from the genotypes in the data set, you can predict the abundance of transcripts. And then when you go back to a large cohort where you've only got the genotypes, you actually don't measure the transcripts. You predict the transcript abundance from the genotypes. And then you ask whether or not there's a mean difference in predicted transcript abundance between cases and controls. So that's called TWAS, and it's very powerful because it gives you sort of summary gene statistics instead of single SNP statistics. And so there's a large literature doing TWAS. And then you can imagine doing similar stuff called EpiWAS, which is epigenome-wide association studies. And that's where you're looking for epigenetic marks that are significantly associated with the trait. But just be careful. The term EpiWAS has also been used, and sometimes that refers to environment-wide association studies, which is where you ask whether or not an exposure such as plastics or omega-3 fatty acids or whatever would be an environmental metabolite you can associate with the trait rather than a genetic thing. So an EQTL is a SNP that influences the abundance of a transcript. So it's basically the same as GWAS, except that the trait, instead of being disease status, is the abundance of the transcript. So that's the topic of a whole lecture tomorrow. And typically those EQTL act within 500 kilobases. In fact, more often they act within 10 kilobases as the transcription start site. An E gene is any gene whose transcript abundance is regulated by a locally acting SNP or cis-EQTL. And so any E genes are good objects for doing TWAS with. A methylation QTL is a genotype that's associated with a degree of methylation at a CpG site. So those beta scores, so the proportion of cells methylated at a CpG site in a sample, those themselves are quantitative traits that we can do MEQTL analysis. And it actually turns out that they're somewhat independent of the EQTL. And yet, in some cases, you get a stronger overlap between methylation QTL and GWAS signals than you do EQTL themselves. Okay. The measure of methylation is this beta. It's just typical. It's just a range from 0 to 1, the proportion of sites that are methylated. A histone mark QTL, a chromatin conformation QTL, and there are many other types like that. Okay, so and then those are all the basis of doing integrative genomic types of analysis. I'm going to spend the last 10 or 15 minutes of my lecture just with my own introduction to single cell analysis. So most of what Peng will talk about from here on out is single cell RNA sequencing. He will go through this on his own terms. I think it's actually useful to hear it from two different investigators. As you know, I'm more of an empiricist, and he's more the machine learning bioinformatician. So we have slightly different emphases. So I'm actually going to start a little bit with the technology. So the idea, this is what's called droplet-based single-cell RNA-seq. 10x Genomics is one of the companies that does it. There are 3 or 4 other companies, but the one most commonly used is 10x. And so they have something called a Chromium controller. They cost about $70,000. And when it comes to your lab, you look at it and say, well, where's the rest of it? Because it's only about the size of a small PCR machine. And what it's doing is that you feed into that cells on one side and a cocktail of barcodes and primers and enzymes and reagents, as well as oil. And as single cells flow through a chamber in the apparatus, they get a missile, a lipid droplet gets formed where the cell is encapsulated along with all of these chemicals and enzymes. And then within each of those droplets, a whole series of reactions are carried out that allow you to profile the contents of those single cells. So the first reaction is reverse transcription, incorporating these barcodes. I'll show you in the next slide. And then from that, you generate libraries. So actually the libraries are made, the cDNA step, the first steps happen inside these droplets, and then they're broken open and you make a full DNA sequencing library out of the contents of all of those cells. And then you take those to sequencing, typically Illumina next-gen sequencing, no longer HiSeq. These days you're usually using something called a NovaSeq or a NextSeq at any rate, so the next version's up. You can also take it to Oxford Nanopore if you want to get full-length reads, and that can be particularly useful for example, characterizing repertoires of immunoglobulins or other molecules you might want to see in their full extent. So these methods are actually just tagging. So they tag either the 5' or the 3', depending on the way the protocol is set up. So it's one or the other. Most often 3' end of the gene. And it's just a single read. So you're not getting the full representation of the exons by any stretch. There are other technologies for doing that. But in these droplet methods, it's just a tagging. So you get all that data, and then you go through all the procedures that Peng will walk us through. Okay, so I mentioned that droplet sequencing is just one of the methods. So there's something also called SmartSeq or SmartSeq2, and that's essentially full-length RNA sequencing applied to libraries generated from single cells. So that's expensive, $30 or $40 a cell. So it's done in 96-well plate format. A typical commercial option is something called Rhapsody from Becton Dickinson. It's very low throughput because you're only going to be looking at dozens of cells per sample because it's so expensive. But the advantage is that it's really comprehensive. So if you really want to get a full readout of exon structure and splicing and things from single cells, this is the way to go. If instead you want to get many, many cells from a large number of samples, then they do this encapsulation in droplets with enzymes and reagents. It's high throughput. In fact, it's now cents per cell, as we'll see in a second. But as I mentioned, it's only tags. And it also has a very high zero count rate. Okay, so it's, and the consequences of that will become clear as we go on. Bio-Rad was the first one. So in fact, 10x is a spin-off from Bio-Rad. One Cell Bio is another company. In fact, I just got a flyer yesterday from a salesperson who visited my lab advertising a form of this. It can be done literally in a test tube in a lab without the need for the Chromium profiler. So we might be looking into that. It'd be a lot cheaper and easier to do. SISG is a version I have to mention on this module because it actually is out of the University of Washington. Jason Jules group. So they've developed a way of doing single cell combinatorial indexing in microtiter plates. It's very high throughput, very inexpensive. They've used it to profile single cells in embryos of various species, in whole worms, whole flies. Very, very prolific assay, but as far as I know, there's only 2 or 3 labs in the world really using it. So it's actually quite difficult to implement properly. But it's there. You need to know about it. Maybe over time it'll become more common. And then the other thing I need to say, I don't have it written down on this slide, but if you're doing ATAC-seq, you can do it on single cells. But actually, sorry, you do it on single nuclei. So the first step is to prepare nuclei out of the cells and then do the attack on the nuclei. If you're doing single cell RNA-seq of neuronal samples or samples you get out of fixed sections, there are ways of doing that, and then it's single nucleus RNA-seq. So you see the term snRNA-seq, that means a single nucleus instead of single cell, and there are certainly advantages to that. All right, so how do the droplets work? So you've got these barcodes. In fact, there are 3 barcodes per experiment, and by far the most expensive step in this is actually making the libraries. So the reagents to create a library cost about $1,400, $1,500 per library. And each library will have a sample index. Okay, so that's because you actually will end up sequencing these samples in a multiplex manner on a lane of a NovaSeq. Okay, so you can put 4 people's single cell RNA-seq assays into one lane, and you know whether or not the sample came from me or Pong or from Karen or from somebody else because it's got this sample index. And then the other 2 barcodes are the 10X barcode, which is literally just a barcode of the cell. So every different cell in that mixture gets its own barcode. So now you can assign reads to that cell. And then there's this thing called the UMI, or the unique molecular identifier. And what that is a marker for the actual molecule that got reverse transcribed and then PCR amplified. Okay. So what that means is if you get, very often you'll get, say 30 reads for a particular transcript, but it turns out that they only derived from 3 different transcripts because 10 of those reads had the same UMI barcode, which means that they were the same amplicon, just reread 10 times. And maybe another 12 of them were from a different UMI, and then the other 8 were from the 3rd UMI. So by doing that, you're actually controlling for PCR biases. So in RNA-seq analysis, UMI is a critical control for PCR biases. Okay. And then since the libraries are so expensive, $1,400, if you like me and you like to do things across dozens or hundreds of people, then actually what we want to do is to combine multiple samples in a single reaction generating the libraries. Okay, so I could take my own immune cells, mix them with Pong's, mix them with a couple of yours, and make a single library. And you might say, well, then if you do that, how are you going to identify which cell comes from whom? Well, the answer is that it turns out that we have genotypes in our RNA. So all of my RNA molecules that are expressed have my genotype, my genomic genotype profile, and Pong's are different, and Karen's are different, and so forth. So you can use the genotypes pretty accurately with something called demuxlet as a 4th type of barcode. Now, obviously, if you're doing 3 different conditions for the same individual, you can't do that because their genotypes will be the same. Okay. So anyway, recapping, sample index is a barcode for the sample. So the individual or the tissue or the treatment, for example. 10X barcode is for the cell. UMI is this unique molecular identifier. We can actually add the 4th barcode. Oh, and then there's another way of doing this, which is to use something called a biolegend cell surface antibody, which is what 10X prefers you to use. So what they are is you can actually tag the surface. So before I mix my sample and Pong's sample, I add a cell surface antibody to mine and a different cell surface antibody to Pong's. Well, it could be the same antibody, but we just add them separately. They have another oligonucleotide on it, and that tags those cells as being belonging to me, and then that gets incorporated into the sequencing reaction. Okay. And then a variant on BioLegend now is to actually use those antibodies to actually detect proteins expressed on the cell surface. And then the proteins, instead of being tagged with a fluorescence marker, which used to be done for flow sorting, it's actually tagged with an oligonucleotide. And then they go into these droplets. So you can actually measure the surface proteins at the same time as the RNA inside the cell and get proteomics essentially and genomics at the same time, or transcriptomics at the same time. Now, in a typical cell, you're looking for about 50,000 reads, which may correspond to something like 10,000 UMI and 3,000 expressed genes, something like that. And most transcripts may have from 1 to 5 UMI represented by multiple reads. So if you only see 2,000 or 3,000 expressed genes per cell, does that mean that's all there is? No, because there's this very high false negative rate. So most cells will have 10 to 15,000 expressed transcripts, but with this droplet sequencing, you're only reading a fraction of them, say 20% or 30% of them. But if you measure 10 cells, then you'll see all of them. So the single cell analysis has to account for this high zero count, or what some people call dropouts. And Pong will talk a lot about consequences of that in his lectures. All right, how much does it cost? Well, if you use a NextSeq, that should be an NextSeq lane, that does about 400 million paired end reads at a cost of about 30 cents a cell. If you use one lane of a NovaSeq, it's called an S1 flow cell, you actually get 3 billion reads, which corresponds to about 100,000 reads per cell if you measure 30,000 cells. And the cost of that would be about $6,000 for us right now, which comes out 20 cents a cell. And if I really bite the bullet and I'm willing to spend $25,000 on one experiment, which we actually do more often than I care to admit, then actually you end up with about 360,000 cells at 50,000. And that turns out to be 7 cents for a transcriptome of a single cell, which is just unbelievable. You can do that, okay? So what read depth do we need? Well, it really depends on the cell type. So 50,000 is sufficient for many cell types, if not all. Some, though, require more. It also depends on the application. So if you really want to find low abundance transcripts, you're going to need higher read depth. But if your focus is differential expression, then you may have a different conclusion. In some cases, you need more. In some cases, if you've got many tissues, you may have less. A typical reaction is going to have 10,000 cells. That's what you aim for in a single 10x chromium library. But if you want to define novel cell types and states, then you care most about the diversity of the cell types in the mixture, and you might sacrifice the total read depth. I just a couple of months ago published a paper called Rigor and Reproducibility, Perspectives on Rigor and Reproducibility in Single Cell Genomics. So I've been somewhat critical about the field. I feel like it's a little bit the Wild West from a statistical genetics perspective at the moment. And so what I've discussed in that paper are 5 aspects of reproducibility that I think need attention. So one of them is just repeatability itself. So there's very few studies that are published have been independently validated, which is our gold standard in most genomic applications. That actually is true of bulk RNA-seq as well. But the problem is that if you have small sample sizes, and given the expense of this, typically people are using 3 or 4 samples of each type, then it's very easy to have hidden covariates you're not aware of driving the results. So independent validation is really a key. So I can point you to many studies of Crohn's disease, for example, where multiple studies have just not given similar results. And that may be just because the design of the individual studies is slightly different. You know, comparing ileum to colon and is not going to be the same. But we need to get to a point where there's more validation from independent studies. Secondly, clustering of cell states and states is the very first step that you do. Anybody who gets into this field, any student, quickly becomes quite frustrated because they find that they just tweak the parameters in Seurat or whatever software they're using just slightly, and they get completely different numbers of clusters. And what do you do with that? Well, that's just the way it is. I mean, that's the nature of the analysis. You've got to keep in mind that it's extraordinary what you're doing. But what I'd like to see happen is people actually report support intervals, just like in phylogenetic analysis, you put bootstrap intervals. There's things like RAND indexes and other things that we should be using to represent how confident we are in our cell type assignments. Because if you divide a data set into two, or you merge two data sets and then you recluster on them, what you typically see is the cells get assigned to the same cluster about 80 to 90% of the time. So there's a lot of ambiguity in clustering that is just an unavoidable fact of single cell genomics. Now, I talked a lot about, in this little mini review, about about the fact that in significance testing, oftentimes you'll get volcano plots that look something like this. So you get a peak of incredibly high significant values, ridiculously high p-values of 10 to the minus 120, despite the fact that you're only comparing 3 or 4 samples. You'd never, ever get that in bulk RNA-seq. And the reasons it's happening is because people are actually treating individual cells as biological replicates when they're not. They're actually technical replicates. So that's a shame. Maybe it doesn't matter. But the other thing I'm showing here is that actually it's not unusual as well to get these sort of double volcanoes where you get a bunch of genes that fit that model and then a bunch that are a lot more variable. But because you've got so much power, these things will be at a cutoff of 10 to the minus 5, the false discovery rate cutoff. There may be a whole lot of significant stuff, but really, if you did the analysis at the single sample level, they wouldn't be significant. Okay, so there is a trend emerging for people to start doing what's called pseudo bulk analysis. So you assign cells to clusters, but having done that assignment, you then pool all of the cells of that type, and then you treat them as a bulk sample or a pseudo bulk sample. And then what it forces you to do is to actually treat those as collectively as your biological replicate, instead of gaining way too much power from the fact that otherwise you're treating them as biological when they're really technical replicates. Okay. Way too many papers to date have failed to fit random effects in their analysis and adjust things with the appropriate statistical model, modeling covariates. So just an example of the impact of that. So down here, consider gene A in 3 different samples, and where the color of the dot is proportional to the intensity of expression. And so what you're seeing here is just eyeballing it. It looks like there's a gradient from sample 1 to sample 2 to sample 3 of increasing expression, or at least sample 1 is less than the other 2. But my point here is that if you've got, hundreds of cells in each of those ones, you'll get a very highly significant result. But actually, if you just transpose randomly sample 1 and sample 2 from those 3 analyses, the mean expression across all of the cells would actually be the same. So I've actually calculated that out. So you'd actually have no difference between the 3 samples if you did that. And really, it's just attributable to a single sample. So way too many studies just actually ignore that inter-individual variability and just pull all the cells from this sample, all the cells from that, and all from that, and they get a result that this sample is different from that one, whereas it really isn't. Conversely, down here, you may look at gene B and conclude that there's actually no difference between the samples because the average expression is the same, given that there's high variability. Oh, and by the way, in these dot plots, what people typically represent is the color is the average expression per cell that expresses it. And then the size of the dot represents the fraction of cells expressing the gene. Okay. So those 2 things together, if you add them up, you'd actually find the same levels. But if you did the proper analysis, adjusting for individual variation, you might actually find that actually there's a significant difference because 2 of these ones are low. Just had the one high one, whereas these ones on average are higher. And that's sort of shown for gene C here, is that overall there would be a difference for gene C because there's high repeatability across the samples. Okay, so there's just not enough of that sort of modeling at this point in the literature. And then my final point is that normalization is all very, very standard so far. We've got a lot of opportunities for doing better normalization strategies to learn more about single cell data. So I'm certainly not saying that the published results are wrong. Some are. It's hard to know which ones are, but statistics are really still in a phase of development and implementation in this field. I'm getting late. I'm going to stop. I'm going to skip over that other slide because one will cover that material anyway. Just to point out, there are different ways of visualizing data. I think Pong will go into these. So TSNE is one of them, UMAP's another. So you get your clusters and then you project them. So both of these methods are producing very high dimensional data. So lots and lots of principal components essentially going into defining the cell types. And then you're projecting those cell types down onto 2 dimensions. And in both of them, the distance really doesn't have much meaning. Okay, so we're typically looking at plots and saying, oh, the distance between 2 clusters must be proportional to how different they are, but that's not the case. They're just visualization tools. And yet they're very, very useful for finding. So in this case, in PBMC, these are all the lymphocytes, these are all the monocytes, and then these are all the B cells subtypes. And then the main group, the software that's used by just about everybody is something called SIRAT from Satija Lab at NYU. Wonderful software. You will all get to know that, those of you do this. This is called anchoring. Okay, so for better defined cell types, we know exactly which genes are markers for those cell types, and you can plot them in your own data set and allow transferability across studies. So in this case, this is defining CD4 T cells, or CD8 T cells, and then these are CD4. Turns out that monocytes also express that marker. All the lymphocytes are defined by CD3D, and so forth. So that's powerful. And then this is a different way of representing the data. I've already talked about dot plots. You can also represent them by these sort of more like flow diagrams. It's motivated by flow cytometry, but you can actually compare your different populations of cells to individual genes. It's more like a frequency distribution than a dot plot. And I think this my last slide is just making the point that there are extensions of this technology. So there's a TAC-seq at the single cell level or the single nucleus level. There's site-seq, is that surface protein sequencing. I really, really like perturb-seq or crop-seq. So these are methods for doing single cell CRISPR mutagenesis and then measuring transcriptomes within the cells where that mutagenesis has happened. Very, very powerful. And then repertoire sequencing is sequencing the T cell receptors or the B cell receptors for anybody doing immunogenetics. These companies offer tremendous tools for doing those types of analyses. Okay, I've gone over by about a quarter of an hour, but that's fine. I actually meant to do that. I'm going to field a few questions and then suggest that we get back at 4:30 for the last lecture of the day from Pong. Okay. Now, put a question in the chat. Yeah. So what happens in pseudo bulk analysis is that you literally, there's a couple of ways of doing it, but the main one is to take single cells. You identify their clusters using the algorithms that Pong discussed and we'll discuss more. And once they're in a cluster, you just assume that all of those cells are from the same state or the same cell type. So we know that there's a very high dropout rate. So any given cell will have so for any given cell type, you typically have maybe 1,000 genes present in all cells of that cell type, and then another 1,000 that are only seen in maybe 10 or 20% of the cells of that type, and then others at lower abundance. So as you pull all of those cells together and you just add up the read counts, you're really approximating a bulk RNA-seq experiment. So if you have 50,000 reads and you do that in 100 cells, you've now got 5 million reads. Okay, so that's like a bulk RNA-seq analysis, and then you can process it just as you would bulk RNA-seq. So I really like that approach, and it's got really good statistical properties because you can now use all the statistics that are available. The issue that some people have with it is that it's dependent upon how well you do the clustering to begin with. Okay, and we've already seen that there's only about 80% overlap in assignment of cells to clusters, even in good data sets. So there's some ambiguity to that. So one way to get around that, there's a nice paper called Vision that came out. There's some software that actually has a Shiny app associated with it. What you can do in Vision is you do your UMAP clusters, and then you make pseudo cells out of all of the closely adjacent cells in that UMAP space. Okay, so you find all of the cells that are very close together, and you make a pseudo cell out of it, and then you treat those, then you fit to the expression in those pseudo cells, those pseudo cells, and then you just regress parameters of interest on those. So it could be all of the terms you're interested in. So you actually don't need to have the cell type defined. You allow for those clusters to be multiple cell types, but you can actually ask in different regions of your gene expression space, are you seeing there's an influence of treatment or disease or whatever it is. So that's software called Vision, V-I-S-I-O-N. I'll look up the paper and put in a chat. So it's a variant of pseudo bulk analysis that I wouldn't be surprised to see gets a lot more play. It's because it's quite new. Yep. Okay, so I should imagine people are getting a bit tired and needing a break. So let's meet again at 4:30, so in 10 or 12 minutes from now.